All right then, on to Chapter 9, Patterns of Inheritance. Uh, One of the things that we talked about with meiosis is how there are so many different ways by which we can create genetic variability in a population. Well, if there are so many ways by which we can create it, is there any way to track it? Is there any way to figure out, okay, what do I expect from the next generation, knowing the generation that I have today? And it turns out there is, because inheritance follows very specific patterns. And this is what we're going to investigate in this chapter. As we already heard, there are a variety of different traits that make up an organism, and these traits reside on chromosomes, of course. Some of these traits are the common kinds of traits, and when it's really common, we call it the wild type. Not in terms of wild and crazy, but the assumption is that the most common feature is probably the one that nature came up with first, and that is what we call the wild type. There are some others that may actually be fairly rare or unusual in any case. So you see this sometimes with color patterns. Most of you probably know what a budgie is, parakeet, right? Those little birds. And most of them are green, but then there are also some that are blue and some that are white. Green is the wild type of a budgie or parakeet, and that's what you will most commonly encounter in the wild. But these other ones also exist, and so you can tell that there is sometimes an additional color that can get into the mix. And so the question is, why does this happen? The reason why we see these differences and why we see certain frequencies in these differences is because of the rules of inheritance. Inheritance means that you are going to pass things from one group of individuals, from the parental generation to the offspring generation, and you see patterns emerge when you do this a lot. And based on these patterns, we can recognize that there must be certain underlying rules about this whole thing. So that's what this chapter is all about. We're working through the rules of inheritance. Now, in order to be able to do this, we're going to have to go over a certain amount of terminology, as we did before. This is always the basic stuff. So remember, the things that we have in boldface print here, those are going to be important. Some of these you already know. The overall appearance of an organism, that's the phenotype, and this includes all kinds of things about the organism that are overtly visible. Things like what it looks like, but also including various aspects of its physiology, how you interact with certain chemicals, for example. Lactose intolerance is part of your phenotype. The characters that we're looking at, the characters are those features that can be passed on from one generation to the next. That means they have to have their basis in genetics, and they have to be variable. And so if you're looking at all the different characters that make up a human being, you'll find that many of those characteristics are clearly passed on from one generation to the next. You'll see people who have grandma's nose or grandpa's ears or those kind of things, and those are ultimately because you are related to grandpa and grandma. There are also features which are not heritable. So if you have a scar, that is not something that's passed on to the next generation. That is an environmental feature. And so as a consequence, that would not be a character that we look for here. The trait is the variant of a character, and what that means is when you're looking at the nose as a character, well, there are things about the nose. You can have a long nose or a short nose. So the trait would then be the length of the nose. The long nose is one kind of trait, and so this is what you'd be looking for in a trait. So character would be the nose, and then the particular state that the nose is in, that would be the trait. An organism's genetic makeup, in contrast, is its genotype. So we have this dichotomy between the phenotype, that's what you look like, and the genotype, which is what caused what you look like. So the genotype consists of all the genes, all the genetic material that ultimately contributes to you and what you look like and how you function as an organism. And when you look for these particular genes, you'll find them on chromosomes somewhere, and the exact position of these genes on a chromosome, those are called gene loci. So one locus, many loci. So that's some basic terminology. So as we navigate around the various aspects of inheritance, we can use those terms. The term genetics itself as a science was something that was coined, about 150 years or so ago. And it was coined in reference to somebody who had figured out some of these important patterns and didn't really make a huge splash at the time. The gentleman who did this was Gregor Mendel, pictured here. As you can see, he lived in the middle of the 1800s. And the reason why he stands out in the annals of science is because he was somebody who really used what we would now clearly identify as a scientific method. He had some ideas, he had some hypotheses, he created some tests, and he wrote everything down and published on it. And so as a consequence, this knowledge became available, even though at the time it was available in German language only. It was not something that was readily accessible in the absence of the internet and the type of communication we have today. And so some of what he put out needed to be rediscovered sometime later, but still it left him with the honor, I suppose, of being the first one to figure some of these things out. The reason why he could do this is because he had an education that at the time was available primarily to members of the clergy, so he was actually an Austrian monk. And as a consequence of that type of job, he not only had the education, but he also had the time to be able to devote himself to these types of studies. And he was able to manipulate the plants in the Abbey garden, and based on all of these experiments, which he conducted over a period of probably 20-some years, he really discovered the principles of genetics. Now, he didn't call it, oh, here I am discovering the principles of genetics. He was just conducting experiments because he enjoyed the intellectual challenge to figure out what was going on with these plants. Now, the plants that he used were garden peas. And garden peas are the kind of things you could grow. And if you were to apply yourself like Mendel, you could redo these experiments. And this is another aspect of the scientific method, that if you clearly outline how your experiments work, you can have somebody else do the same experiment and come up with the same kinds of results. And so when you study garden peas, you find that just by chance he had an organism that was very appropriate to study genetics, because peas, it turns out, have some easily observed traits. There's quite a few of them actually in a pea. Plus, peas are also easily manipulated. And so when you're working with an organism that you can actually conduct experiments on, then of course that increases your ability to steer the experiment in a certain direction. And then lastly, peas normally self-fertilize, which means that an individual does not go outside of its own identity to create offspring, but they are basically clones in how they reproduce. And when you look at what that pea plant looks like, you have here in the center, this is a carpel, this is the female part. And then you have around it, you have various of these male parts called stamens. The stamens make pollen, and in the pea, normally, here you have one of the petals folded back, but if the petal stays closed, then you would find that these pollen-producing parts here would release their pollen right onto the carpel, and the carpel would then produce the eggs and the seeds, and you'd end up with self-fertilization. This is essentially a closed system. Now, if you turn it into an open system, then you can manipulate this. Rather than allowing it to self-fertilize, what if you cross-fertilize? Cross-fertilize versus self-fertilization means you now don't have the same organism fertilize itself, which in the case of these plants can happen, but you actually involve multiple individuals. And of course this makes it interesting because it's an experiment to see what happens if I have two different ones of these pea plants, perhaps displaying different key characteristics, mate with each other, and then see what the offspring do. And that's basically what Mendel did. And so his experiment ultimately consisted of series of treatments in which he cross-fertilized plants that had different traits that he was interested in. So you look at one trait at a time and you just figure out how does the offspring differ from the parents, and then what happens to its offspring and its offspring and so on. And of course if you do this long enough and often enough, then you can end up with some interesting data. He started with true breeding individuals, true breeding for the particular trait that he was interested in. So true breeding in this experiment here that we're looking at would be for white flower color. So the character we're going to look at is flower color, and the trait is going to be white versus purple. So true breeding individuals means that this is a set of individuals that have only ever self-fertilized. And so since he was in charge of this garden, it would be fairly easy for him to figure out, okay, over there in the corner is where all my white flowered individuals are, and over here in the other corner is where all my purple flowered individuals are, and they're always white and those are always purple, and so those are my true breeding individuals. And those are basically the parents of this whole business. And then he would bring those together and cross-fertilize and then figure out what happened to the offspring. So once he does his experiment, then he simply counts the new pea plants after he plants the peas. So the picture here involves, first of all, to remove the stamens from the purple flower. You remove the stamens so that self-fertilization cannot occur. So before this plant even matures, you're going to open it up and you're going to clip off all the things that might produce pollen, and you're eliminating them. So that means now this plant can no longer self-fertilize. Then what you're going to do is you're going to take some of the pollen from the other color plant, and you can use that with a little brush, and you brush some of the pollen grains from the white flower onto the purple flower. So now you have the male parts from the white flower interacting with the female part of the purple flower. And so you're mating, basically, the white flower with the purple flower. And so in order to do this properly, you make sure that you use flowers that have been closed, that haven't been opened yet. You can do this multiple times, and whenever you do this, you have a parental group of organisms, the pure breeding white flowered ones and the pure breeding purple flowered ones, that are going through this exercise. Then of course peas do what they will. They mature eventually into the fruits, peas in the pod, and of course you can't necessarily tell by the actual peas what color the flower is going to have. So what you have to do is you have to take these, plant them, and then wait some weeks, and eventually you're going to get new flowers, and you see what happens, and then you have your offspring. This offspring is what started to give him some pretty interesting results. So here are some of the patterns he observed. When we're looking at these kinds of crosses, and this is what we're doing, crossbreeding plants, we start by looking at each character individually. And so because we're looking at each individual character, we call this a mono-hybrid cross. Hybrid is this process of cross-fertilization. It's taking two different things and fertilizing them together, and so that's what we call the mono-hybrid cross. And in particular, we're looking only at the one trait. So these plants are all identical. The only thing that they're differing in is whether it's going to be a white flower or whether it's a purple flower. And so Mendel set up his experiments like this. The pea generation, P is always going to be parental. So the parental generation is critical to the setup. And in this case, the parental generation is going to be either plants with purple flowers or plants with white flowers. And these are true breeding, means that in this group you only ever find purple flowers, and in, whoa, I just pushed something unusual. And so you only ever find purple flowers in the one, and then only white flowers in the other. Secondly, you count what the offspring look like. And these offspring are called the F1 generation because back then people did a lot with Latin, and so this is the first filial generation. First filial generation. Filius in Latin means son. Filia means daughter, and so as a consequence this is the first filial generation. And this is the first real result of this, that when you do this, it doesn't matter how many times you do this, all plants have purple flowers. 100%. Now that's obviously very interesting. I mean, if you have something that's purple and something that's white, where does the white go? Well, obviously the white disappears, right? And so you may, as your first partial conclusion, say that white somehow disappears, it's weak, it doesn't work, whatever it is, you have a problem with white, clearly. Now, what Mendel did is then he took those F1 plants and fertilized them with each other. So now you take purple flowers that he knew were part of this first filial generation and mated them together, and you end up with the second filial generation. And the second filial generation was remarkable because suddenly the white reappeared. So this is interesting, first of all, because the white obviously somehow skipped a generation. Secondly, when you look at the numbers, and he did a lot of these experiments, you end up with a very specific ratio. 3/4 of the plants end up with purple flowers, and 1/4 of the plants have white flowers. Now, when in nature you find clear-cut numbers like this, 3/4 and 1/4, then that tells you that something is probably not random. Right? I mean, there's some underlying rule here. We don't know what it is. Now let's think about this for a minute. Really think about it. If the white disappears and then it reappears, how is that possible? Just in terms of the color itself. Well, one way by which this can happen is that in this generation, white spontaneously appears. Right? There it is. Ta-da! It's come back. And that's true. It could do that. The problem with that is that if it's some chance recurrence, why do you end up with exactly 25% of the time? That's a little puzzling, because if it's by chance, then you would say, well, sometimes it's more, sometimes it's less, right? So that doesn't make sense. The second thing that could happen is that even in this generation, even though all the plants are purple, maybe the white is hiding somewhere. And so if it's hiding somewhere, that means it is somehow being carried forward, but then you have another problem. Where would it hide? I mean, either it's there or it's not, right? How could it possibly hide? And so somebody like Mendel would look at these results, and after having done this for some years, you really have to sit in your thinking chair and think and try and figure out how this can possibly make sense. And so all of this experimentation and hypothesizing and so on boils down to trying to find a solution to this. And Mendel was pretty astute when it came to this. Well, first of all, when you think about these results, you come up perhaps with additional hypotheses, with additional ideas about how this might work. And so Mendel ultimately came up with 4 hypotheses about these traits he was interested in could be inherited. First off, he would have to say that there is something on these chromosomes that transfers from one generation to the next. Now, he didn't know chromosomes. He may have called it pieces of information. He may call it information that carries over, but whatever it is, today we call these alleles. And so in this case, what we would say is that there is a gene for flower color, and this gene comes in two versions, two alleles. There is going to be a purple allele that makes the flower purple, and there's going to be a white allele that makes the flower white. So there are two pieces of slightly different information that make flower color. The second thing is the only way by which we can hide the white is by carrying it forward. So whatever that white parent gave us, it's still there in the second generation. But the only way it can be there is if the purple goes together with the white somehow. And so you can't do that by only having purple move from one generation to the next. The white also has to move. And therefore, he decided that in order to do this, you must be able to carry one piece of information that's purple and another piece of information that's white. You must be able to bring both of these along, because that way you get the white for sure. You can't just lose it somewhere. Even though the whole thing looks purple, the white must still be there. And I deliberately drew the white in a slightly smaller circle because the white isn't visible, right? It's not visible. But if we're going to encounter it again in the next generation, and we do it at this 25% level all the time, then we're going to have to carry it with us. And so what he said is that if we're going to do this, then one of these alleles is going to be dominant. Dominant means it is stronger in some way, shape, or form than the other one. So we would say capital P for purple, that's going to be dominant. And we would say in our genetics speak, if we use little p, that's going to be the white. And the reason why we use the little letter, the small letter, is because it is not dominant, but it is recessive. And so when you look at inheritance like this, and one of these traits completely disappears in one of the generations, then that trait that disappears, that must be the recessive one. Recessive in the sense that it recedes into the background for a while, but then it comes back. And so this is what Mendel started to see in these flower colors, that this is how it has to be if we're going to go from one generation to the next with the patterns we have. And it's a very exciting discovery, because ultimately you're going to be able to track what happens in the next generation and in the next one and the next one. I mean, it's like figuring out how life works a little piece at a time. The last thing he thought is that if I'm going to have information transfer from a parent to an offspring, which clearly is the case, because the purple is going to produce purple, it's not producing orange, it's not producing red, it's going to be purple. And then in the next generation, it's not going to be orange or red or anything else either. It's either going to be purple or white. So we're back to the parent colors. So this information is preserved. It just travels in certain ways. So if it travels in certain ways, is it okay? So if it travels in certain ways, then it means that you can really only have each gamete, this is what we now know, each egg or sperm cell, carry only one of these alleles. So if you have an egg cell, maybe it carries the capital P. And if you have the sperm cell, maybe it carries the little p. Something like that, but not both. Only one or the other. And that's the key thing. So now you've put together all these different rules, and these are initially hypotheses, and now you try and figure out whether this works in real life. The person who actually came up with a way to test for this is this guy, Reginald Punnett. And as you can see, when he lived, he came into his academic career substantially later than when Mendel lived. So Mendel's findings were literally unrediscovered or unexplored, even though he'd published for some years. And even though Mendel, you can still go to where Mendel has his lab notebooks, and where in fine handwriting he explains all these things and so on. It's pretty interesting to do. And so what Punnett did, he formalized this proof. It's kind of a little bit like a mathematical proof, but he put all these things together. So he said, well, here we are. This is the parental generation, and normally in most of our cells we have cells, and in these cells we have 2n chromosomes. Remember that from chapter 8? 2n chromosomes. So therefore we would have one chromosome with the capital P on it and the other chromosome with a capital P on it. Likewise, we have the same thing with the little p. Now the little p, if you remember, that's recessive. The big P is dominant. And when these produce gametes, this is where meiosis comes in, we end up taking only one of these and putting it into an egg or a sperm. Only one. That's where meiosis comes in. Now I'll show you more about this later, but this is where you can already see the effects of meiosis. You have a reduction division from 2n to n. So all your gametes are going to be n, whereas the parents are going to be 2n. Now comes fertilization. You take the n from here and the n from there, and you're going to make 2n again. But in this case, you have the purple allele and the white allele. Purple is dominant, which means everybody here is purple. Right? The capital P literally overshadows the little p. So even though the little p is here, you can't see it. And so the dominant overpowers the recessive. Now when you then produce gametes from this, there are two options now. You can either produce the capital P, if that one goes into the gamete, or if this one goes into the gamete, you can produce the little p. And because neither one of these is better than the other, it's just 50-50. Depends on who just happens to go into the egg or the sperm. It's that game of chance that we talked about. Now, once you have this situation, you actually have to make a little table, and this is what we call the Punnett square. So this business here, making a little table, is the Punnett square. So on the one side, you're going to put the contributions from mom, from the eggs, and with mom it can either be a capital P or a small p. On the other side, you're going to put the information from dad. That's the male. This is the female. And you're going to end up with the same, but on the other side. Now you multiply them together. The capital P and the capital P together, well, that obviously makes two capital P's. The little p and the big P, that's big P, little p. And here again, the big P and the little p, capital P, little p. And then down here, little p, little p. So suddenly you realize where these numbers come from. In 3 of these cases, it's going to be purple. 3 out of 4 boxes in that Punnett square. Well, 3/4, that's the number we had, wasn't it? Only 1 out of the 4 is going to be white. And there's the reason why we end up with that 1/4 number. It's because of how these alleles work together with each other. And that's what you end up with. You end up with these ratios, and these ratios never change. This is one of the fundamentals of how information is inherited. You end up having, in this case, 3 of the boxes with purple individual flowers and 1 with white. So what Punnett did is he took the information from Mendel's research and put it into this setup, which was subsequently named after him, and said, hey, this is how Mendelian genetics works. This is why he was right. He took those numbers and actually made sense of them. Now, the phenotypic ratio is what I just showed you. Phenotypic ratio is actually looking at the colors. And then the genotypic ratio is a little bit different because here you have only capital letters, only small letters, and then in both of these cases you have the mixed letters. Only capital letters, only small letters, and the mixed letters, there's two of those, right? So one box with capital letters, one box with little letters, and two boxes with mixed letters. And so the genotypic ratio is 1 to 2 to 1, whereas the phenotypic ratio is 3 to 1. And so this is how the various aspects of Mendelian genetics were put together. This took some time, it took some doing, and people were just starting to look inside cells and to actually look at chromosomes and figure out how those work. And that's one of the things we'll talk about a little bit later. So that is part 1 of inheritance.